Hey guys, so I'm going to be talking to you today about viruses and what they are, what they do, what they look like. First thing you guys need to know is what is a virus. And a virus is a non-cellular infectious particle of protein and nucleic acid that replicates only in a host cell. So, as you can see on the screen, these are different types of viruses, along with this one. So, different types of viruses are stuff like the flu, hepatitis, smallpox, influenza, herpes, HPV, and the West Nile virus, Zika, and swine flu, just to name a few. So, pretty much the first type of bacteria we're going to talk about, or virus that we're going to talk about, and the most common one we see is the bacteriophage. So, what this does is it's a very vital part in the virus's replication. And what a bacteriophage is a virus that infects other bacteria. So in this, you can see this bacteriophage attached to a new bacteria cell that it wants to host and inject the DNA into. Here's the structure of the bacteriophage. As you can see here, this little crystal head that it has is the protein coat, which is pretty much the protective coat of the vital parts of a bacteriophage, which holds the nucleic acid, which is this purple slime gel sac you see, and the DNA, which is this blue string that's inside the nucleic acid. So you go down a little bit, you see the tail or the sheath, and these little leg things are called the tail fibers. These little points right here are what help grab onto the cell that it wants to inject. So here's a little diagram of a bacteriophage injecting a new host cell. So it starts off with the landing. Okay, it just lands and sits on top of the cell membrane. It will then squat down and begin to attach itself to the membrane of the cell. From there, it will then start to inject into the cell, and it will put the DNA into the new host cell. From there starts the process of the lysogenic cycle or pathway. So, as you see, the bacteriophage will land on the new host cell. It will inject its DNA. From there, the DNA will go into the nucleus and begin to produce more bacteriophage. After it produces enough, it will then release all of the bacteriophage that it created to start this cycle over and over and over again, which causes more viruses. Now, the next thing that you need to know is viroids. And what a viroid looks like is this. Now, viroids are small, non-coding infectious RNA. So pretty much where you see these most is in plants, such as potatoes, tomatoes, avocados, apple, coconuts, stuff like that. And what it does is when it infects a plant, it will begin to stunt its growth and deform it, as you can see right here with this potato plant. After that, you will see pathogens. Now these are examples of some human pathogens. Now what these do are they're disease-causing agents. So anything that can carry disease is a pathogen. So ticks, fungus, parasites, other type of viruses are example of pathogens. Now, another type of virus that y'all need to know is the retrovirus. And what a retrovirus is a virus whose RNA is used as a template to produce double-stranded viral DNA within a host cell. So this is the process of a retrovirus attaching to a new host cell. Much like the bacteriophage, it will land on the cell membrane and infect it with its RNA. When it infects with its RNA, it will go into a process called reverse transcription, and it will begin to make the new double-stranded DNA. From here, this DNA will go into the nucleus, and begin forming and attaching itself to the DNA that's already being produced by this cell. This will begin the reproduction process of the new cell. Here, mRNA will go out, which will turn into viral RNA, and it will then be assembled and released as the new retrovirus. Now, example of a retrovirus would be HIV/AIDS. Now, the next thing that I'm going to talk about is the common viral diseases. Now, these include measles, mumps, smallpox, chickenpox. What else? That's pretty much cold sores and genital herpes are all examples of common viral diseases. Now, a disease vector is animals that transmit a pathogen from one host to the next. So examples of this would be mosquitoes, pigs, rats, anything that can carry a disease and can be transmitted. What these do is, for example, when a mosquito were to bite a human, it can then transmit the disease, malaria, Zika, stuff like that. Pigs can transmit the swine flu, and rats just carry a bunch of viruses and diseases, not too many to name. Now, the next process that we need to know about viruses is viral restorement. Now, viral restorement is when two related viruses infect the same individual simultaneously and swap genes. Now, example of this would be in 2009 when the H1N1 flu appeared. It had the mixture of the human virus and the bird flu, and it was then called the swine flu, which is a perfect example of viral restorement. The next two things we need to talk about is an epidemic. An epidemic is an outbreak of diseases in a limited region. Examples of this would be the swine flu, malaria, the Ebola virus, the Zika virus, and etc. A pandemic is an outbreak that happens simultaneously throughout the world. Luckily, there are not any really big cases of pandemics happening in the world due to our good healthcare. So thank you guys for watching. I hope you guys learned a little bit about viruses, and thanks again.